theoretical aspects, although very applied. And for the second half, Justin is actually going to walk us through preparing a protein for simulation in Nandi using VMD. So that'll be the second half. Justin and I are actually both from Minnesota, so our undesirable delicacy is lutefisk. So we're going to put the lutefisk up front in this talk and a little bit more. And after learning to use VMD this afternoon, tomorrow afternoon you're actually going to be doing a wonderful hands-on which has been prepared by three of actually our younger graduate students in the group, Elizabeth, Marcos, and Tim. They've put a lot of work into that. They were actually up all night finishing it off, proofreading. So you should have a wonderful experience preparing the protein, simulating it in Nandi. And one very important point is don't worry about sketching down every last thing that I say during this hands-on. In fact, the tutorial tomorrow afternoon is very complete. That's where you will learn how to do simulations using NAMD. This talk is merely an overview. So PDB, PSF topology, and parameter files. Klaus introduced the concept of a PDB file. That's the most basic file that you can load into VMD, and it's also the starting point for most simulations. These are the files that are deposited into the protein data bank by crystallographers, and you need to start your simulation with some coordinates. If we were doing simulations of water or argon gas, you could make up your own initial conditions. We really need a crystal structure here. The PDB files that you can download from the protein data bank, if you inspect them in a text editor, contain a couple of pages of records at the beginning of the file, which include the species and tissue where the sample was obtained, who did the work, citations for the papers where it was published, the sequence of the protein, information on the secondary structure, etc. All VMD really cares about, and all NAMD cares about, are the atom records in the PDB file. And those contain very basic information, the names of the atoms, which are standardized up to a point, although there are some small differences between conventions which cause some problems that Justin will discuss. The residue names, very standardized, the index of the residues, the coordinates of the atoms, which is the primary data in the PDB file that we care about. An occupancy field from 0 to 1. Sometimes you have different locations for the atom that form the crystal structure. Both will be represented, and the relative occupancy of those locations will be specified. Temperature factor, or beta, as it's referred to in VMD, and a segment ID. PDB files from the protein data bank do not contain hydrogen atoms because hydrogen atoms do not typically show up on crystal structures. However, the force fields that we use for simulations require hydrogen atoms, and VMD provides facilities for adding those to the PDB file. When you're doing a simulation, the only information that comes out of the PDB file itself is the atomic coordinates for all of the atoms in the simulation, including hydrogens that were generated in VMD. Everything else that's specific to the simulation that you're doing is contained in the PSF file. PSF file stands for protein structure file, includes every atom in the simulation and all the atom-specific values. So in addition to the ones that are copies of the information in the PDB file, you have the mass, the type of the atom, which is very specific to the force field, and the partial charge. While every carbon atom in a simulation will have the same mass, the partial charge is dependent upon the specific location of that atom within the residue within the molecule. The only things, as I said, that are not in the PSF file are the coordinates, which are in the PDB file, the velocities, which are essentially random. Initially, they're generated from a Boltzmann distribution, and the specific parameters of the force field. And those aren't in the PSF file because they're contained in a separate parameter file, which is used for all simulations involving the same force field. Also in the PSF file, in addition to the names of the atoms, we need to know what their connectivity is. So every pair of covalently bonded atoms is listed in the PSF file. But there are also other terms which are used to apply forces in the simulation that have to be specified. Angles and dihedrals. So every pair of bonds forms an angle. Every set of 4 bonds forms a dihedral. And finally, a small number of additional terms. If you have a set of 4 atoms that form a planar group, in order to constrain that group to a plane, you need something that's called an improper. And we'll discuss that a little bit now. So how do you generate all these terms in the PSF file from a PDB file? We use what's called a topology file, which is a blueprint for building the PSF file. For every type of residue that is known in the force field, this is alanine, for example, the topology file contains the atom name, type, mass, and charge, and all of the bonds, including bonds between the previous or the next residue in the protein chain. And it also explicitly includes any planar impropers, although those are relatively rare. Angles and dihedrals are automatically generated for all combinations of bond in the molecule, and if you get bored at some point during the second half of this slide, you can try and figure out how many dihedral terms you would actually need even for this simple alanine residue. Also in the topology, there's things called patches, and patches are modifications to other residues existing in the structure. So patches are used for terminating protein segments, joining 2 segments together, modifying the protein state, for example, of histidines, adding disulfide bonds. If you're building PPTI, there is a disulfide bond that you'll want to generate. And in constructing nucleic acids using the Charm 27 force field, the default nucleic acids are ribonucleic acids, and if you want DNA, you have to go through and explicitly deoxygenate all of the residues. So if we have a PSF file that we've generated using our topology file, we have coordinates that we've completed using VMD in our PDB file. Now we need the actual forces and energies that are associated with that. The charm potential function is very simple. As you can see, it has basically one harmonic term for every degree of freedom in the molecule. Dihedrals are the most flexible terms in the force field. For example, rotations take place there, so those have a continuous harmonic term allowing 2, 3, or 2 or 3 different minima. And then the non-bonded potential is split into the familiar Coulomb electrostatics term based on the partial charges that are assigned to each atom in the PSF file. And the non-bonded terms, the representation of the van der Waals interactions is by a traditional Leonard-Jones potential. And this has 2 parameters as well, the depth of the well, epsilon, and the equilibrium distance between 2 atoms. But of course, we need all of these parameters, which is where the parameter files come in. Parameter file is closely tied, of course, to the form of the force field that it's providing parameters for, and to the specific topology file that was used to generate the PSF file, because, of course, the atom types in the PSF file are used to look up the particular parameters. So for every pair of atom types that can form a bond, you have equilibrium values and spring constants. The same for angles, dihedrals, and impropers. Although for dihedrals and impropers, there are so many possible combinations that wildcard atom types, which simply show up as extras, used, so that by only specifying, for example, the 2 central atoms in a dihedral, you can cover an entire class of interactions. And then for every atom type, there's a single van der Waals radius and well depth. But since we actually need these for every pair of atom types, because they're non-bounded interactions, there are simple rules for calculating the pair radius for interactions between two atoms and the well depth for every interaction. Okay, no more lutefisk. We're now into the wonderful world of molecular dynamics. We're going to start out in paradise and get consecutively more miserable as time goes on until we drop over into Justin's presentation. So we've got this potential function that's been parameterized at great length by many researchers over the years. We take this energy function, we differentiate it, we have a force, and we can apply Newton's second law, F equals ma. And in principle, this gives us a set of 3 times the number of atoms coupled second-order differential equations that we can propagate forward in time. Classic Newtonian mechanics. Initial coordinates are from the crystal structure. We can take velocities at random from a Boltzmann distribution and run forward forever. Life goes on. Now, of course, for statistical mechanics reasons, we need to know what temperature we're running at, and we can maintain the temperature of the simulation by adjusting velocities. How should we do that? Well, anyone who's seen any molecular dynamics text is familiar with, first of all, the formula for calculating temperature. Simply the kinetic energy of each atom divided by its expected energy at a certain temperature. Ignore this T. Kinetic energy divided by 3 times the Boltzmann constant, averaged over all the atoms in the system, is our estimate of temperature. So one thing we can do is we're running, measure what the temperature of the system is, rescale the velocities so that the temperature agrees with our target temperature, typically 300 Kelvin. Very simple. If you want something smoother so that you don't have these drastic jumps in kinetic energy during the simulation, you can use a continuous process. And we do this by modifying the equations of motion. So the first 2 terms, simple F equals ma, and add a temperature-dependent friction term so that if the temperature is larger than the target temperature, the velocities of the atoms are slightly damped. If the temperature is too small, the velocities of the atoms are slightly increased. However, it was discovered that if you apply these methods long enough, they will slowly drive energy from the higher frequency normal modes in the system, such as bond and angle vibrations, into lower frequency normal modes. Well, what effect can that have? This is an actual data from a simulation done by a Nandi user. And he was confused as to what was going on in his simulation, in this case pure water. Time was passing. As time passed, at one point he was following the radial distribution function, and things seemed to go haywire, and he didn't understand. Well, I plotted the energies of the simulation, and he was doing this with the temperature coupling method we just talked about. So the kinetic energy stayed the same. However, during this transition, the total energy of the simulation dropped dramatically, and most of the drop was kinetic, some van der Waals increases to balance this out. What's going on here? Well, you can't see that very well from the audience, but if we zoom in on the kinetic energy, you'll also notice that the fluctuation drops dramatically. And as you'll learn from Johan's talks tomorrow on statistical mechanics, the fluctuations should be proportional in some sense to the temperature of the system. Well, if we're measuring the temperature based on kinetic energy, we're getting a different result. Finally, if you look at the volume of the amount of water in the simulation, you see this interesting behavior. That's a phase transition. what happened is the lowest mode in any system turned out to be translation. So all of the energy, kinetic energy in the system had been translated into translation because the initial velocities were non-zero. Now, one thing, and the rest of the modes had frozen, so you had a flying ice cube. And what happened? One thing you can say as well, of course, if you reset the momentum of the entire system to zero periodically, you can avoid this. But really, you know, it's sort of a, if smoking is bad for your teeth, imagine what it's doing to your lungs kind of thing. If this is, you know, an observable thing, Lord knows what other properties of the system are being affected. So how can we avoid this? Well, another problem with temperature control methods that take the temperature of the entire system and adjust the velocities in bulk is face the following situation. You've joined a protein that happens to be highly minimized, it's at 200 K, maybe, with a slightly warmer water bath, maybe 350 K. Would you prefer to heat the water, cool the protein, or do none of the above? Now, of course, as the simulation runs, heat will transfer, and these should become the same. But if you have a temperature control method, you can have the temperature of your system reading 300 K evenly with this kind of a situation. To deal with each atom separately, it's difficult to calculate the instantaneous temperature of an individual atom, but we can use statistical mechanics and Langevin dynamics by balancing a small constant friction term. So we're always subtracting a small amount of energy from each atom and apply random forces that at the same time add a small amount of energy at each time step, so that statistically over time you'll end up with a constant temperature and at the same time avoid some of the problems that can happen with other thermostats. So Clef started out the fairy tale discussion today. So here's a very simple schematic of what we're going to do in any molecular dynamics simulation that you'll be running this week. We start out with initial coordinates, and those initial coordinates are not necessarily all correct. If hydrogen atoms have been constructed in a bad manner, basically guessing based on the other atoms in the system, you may have bad contacts. They may be closer than their van der Waals radii together, which results in a very high energy. Also, you've built solvent around the system, lipids, you may have interference there. So the first thing that you have to do in any simulation is to minimize the system to eliminate those bad contacts. All minimization can do is find a nearby local minimum. And that minimum is not going to have the same energy as, for example, perhaps the true ground state of the protein. But we drop down. Once we're in that state, we can start heating the system and equilibrate the system. And as we add energy through the dynamics, the system should be able to escape any local minima that it's in that have sufficiently low energy barriers. We then end up, after being fully equilibrated at 300 Kelvin, in a sampling of the thermally accessible states in a local well. And from this, you can get all of the short-time thermodynamic properties that you'll be learning about from analysis, look at variable motions in the protein. However, if there are barriers that are nearby, there are other interesting states that play an important functional role. For example, if there's the binding path of a substrate into the protein that contains several intermediates with very slow transition rates, a normal simulation will not sample those in a reasonable amount of time. And this is why the steering forces, steered molecular dynamics that you'll be learning about on Wednesday, are useful because we can then force transitions into these other states, sample into the other states, and experience the entire wonderful world of biomolecular physics. Okay, so that's what you would do in an ideal world where you have this energy function, computers are infinitely fast, and all you have to do is type all of this into Mathematica, say integrate, and go. Unfortunately, there are some details that we need to worry about. First of all, there's solvent. If you've read about the structure of water, water is one of the most difficult substances in the world to simulate because it forms a network of hydrogen bonds. Has something like 20 different 20 different phases of ice that can form different pressures. If you put the system in a bubble of water, at the interface between the liquid water and the vacuum, these hydrogen bonds have to form inward, and you get surface tension, which we're all familiar with from any time that it rains. And to reduce that surface tension, you can form a sphere, and you develop very high pressure inside that sphere. Why would this matter to us? Well, this is a initial structure of F1 ATPase, a simulation that was done in the group over a year ago. And one of their questions was, well, what if we just surround the system by a nice oval of water? What happens? Well, as they equilibrated it, the water bubble deforms towards a sphere, and because of this, the secondary structure, which is relatively delicate on this protein, is significantly deformed. So how can we avoid this? Well, your goal is to say, instead of this small bubble of water immediately around the protein, what if we're in an infinitely large bath of solvent, an infinitely large bath of water? Well, we can provide this illusion so that there aren't any surfaces by saying, okay, we have an internal cell, and we surround the cell on all sides, 27 different sides, by identical copies of itself that it interacts with. And if one atom leaves the cell on one side, it emerges on the other side, and you interact with all of these neighbors. And that way you appear to be in an infinite body of water. So this is what was done with ATPase, and this is actually the periodic cell for the simulation. As you can see, there's exactly one copy of the protein inside the cell surrounded by reasonably sized buffer regions of water. Now this is a huge protein, so this is probably on the order of 10 angstroms of water on the side. So you'll be doing this tomorrow afternoon as well. So if we've got this cell of water, this periodic cell of water, the goal is to eliminate surface effects. As you can see here, the system is completely filling the periodic cell. But since we're dealing with a liquid, liquids have very highly defined densities. And what happens if we, for example, make the volume of the cell slightly larger than the correct value? Well, in very short order, the cube that you see here, and this is actually colored by distance from the center, so you can see the corners on the cube. The corners get clipped off, and since this is a periodic cell, what's actually going on is there is a bubble of vacuum that is formed at the corners of the periodic cell, while the rest of the water in the cell is at its proper density. So instead of minimizing the surface area of a bubble of water, you've minimized the surface area of a bubble of vacuum. How do we avoid this? Well, you have to use constant pressure simulations. You have to set the pressure of the system to be a fixed value. Pressure is controlled in the ideal gas law by volume and temperature. That applies even to liquids. If you have a non-periodic system, you have a pressure of 0, other than the surface effects. However, for the periodic system, you have to adjust the volume of the system. So what any pressure control algorithm uses, and you'll be using one called Langevin piston tomorrow, is that if the measured pressure in the cell is too high, you expand the periodic cell. If it's too low, you shrink the periodic cell. And you don't just change the size of the walls, you rescale all the coordinates within the cell at the same time. And you will get a little bit more experience with that. Going forward, so as I said, that's in the real world where we have to deal with solvent, pressure, non-ideal systems, unlike the cartoon representation. When we have to deal with try and solve these equations on an actual computer, there are a couple other issues which come up. We have to deal with finite time steps. So instead of smoothly propagating the system forward, we have a defined time step, which is delta t. The derivation for the propagator, the integrator that's used, is actually deceivingly simple. If we take a Taylor expansion of the position one time step forward from the current time, just based on the velocity and acceleration, force divided by mass at the current time, and we do the same thing backwards in time, add those together, we end up with a very simple formula that simply says, well, twice the current position minus the past position plus the current force acceleration times the time step squared. That's all there is. Well, this looks simple. This is actually a much better integrator than you would think. First of all, the next term in these sequences, like the velocity, cancels out. So this is actually an additional order of accuracy than you would expect. It's also time reversible, and it's symplectic, which guarantees that if the time step is small enough, you will conserve energy over a long simulation. And you'll be hearing more about this next week from Professor Skeel. Now that time step, if you look at all of the processes that can occur in a biomolecule, you cover 15 orders of magnitude. That time step has to track the fastest process in the system, which is stretching of the interatomic bonds, those simple spring constants. And because of that, we're faced with a time step of 1 femtosecond. And that's actually constrained by the vibrations of hydrogen atoms, because those are the lightest atoms in the system. So we're stuck with a 1 femtosecond time step, and it takes 1 million time steps even to simulate a nanosecond. So that can take a while. There are some approaches that we use in simulations to cut the time down. First of all, doing the non-bonded interactions, those van der Waals and electrostatics terms, you have to do every pair of atoms. Well, as the system increases in size, you have to do n squared the amount of work. Double the size of the system, 4 times as much work. If we take those non-bonded interactions and say, well, they're pretty weak as you get further out, maybe 10 angstroms, 12 angstroms is all we have to go, we can reduce that to order n. So if you double the size of the system, you double the amount of work you have to do. Unfortunately, if you decide to double the size of the cutoff, you have to do 8 times as much work. So large cutoffs aren't necessarily the answer. Fortunately, for the electrostatics forces, which are the most significant long-range forces in the system, there's a method known as particle mesh Ewald, which can cut that down to order n log n time. And it's actually a very minor cost compared to the cutoff calculation. So particle mesh Ewald is a method that you should recognize and use. On the other hand, if we can do force evaluations less often by extending this time step, that'll also win. Some simple methods for doing that are, first of all, if we look at those bonds to the hydrogen atoms, well, rather than letting the hydrogen atom oscillate, we can just fix the length of that bond. And if we do that, we can take a 2 femtosecond time step. Also, these long-range electrostatics forces, because they're very weak, they don't change very often, so we can evaluate those less often. Now, as soon as you apply these methods and you get your simulation down to size, time catches up with you. And over time, we've seen from BPTI that you're going to be working with, even in 1996, an atom system 10 times that size. And a year or two ago, a 300,000 atom system. And even more recently, there's been a 2 million atom simulation done with Nandi of the ribosome. So systems are only getting bigger. And even these systems are not gratuitously large. In fact, this is only half of ATP synthase. There's another subunit missing. And this is only the DNA binding portion of this receptor. The entire estrogen receptor domain has been removed to make the simulation small enough. So how do we deal with this? Well, that's where Nandi comes in. Parallel computing is really the only answer. This is a 3,000 processor terascale system called Lemieux at the Pittsburgh Supercomputer Center. And on one processor of the system, for that very large simulation on the last slide, it takes 28 seconds per 1 femtosecond time step. With Nandi, we can use 1,000 processors on this machine and cut that down by almost a factor of 1,000. In fact, a factor of 750, which is 76% efficiency, which is pretty good. And we've actually, we're given a Gordon Bell Award in supercomputing last week recognizing the scalability of Nandi. Unfortunately, with 90-some participants and only 150 nodes at NCSA to use, you won't be able to experience this week. But if you do ever get in the situation of modeling a very large system, it's good to know that this is here. A couple tips on Nandi. If you're going to use these large systems, when you're running on 1,000 processors, you can waste computer time very effectively. So these are some things that you may or may not want to do in an Nandi simulation, depending on your goal. First of all, as I said, if you double the size of your cutoff, you're doing 8 times as much work. Particle mesh Ewald is order n log n, very efficient. So if you want to waste time, by all means, use a 16 angstrom cutoff and ignore particle mesh Ewald. Also, if you have a small system, you can use as many processors as you want. But if you're using more than one processor per 1,000 atoms, the simulation won't actually run any faster. There are just limitations on any parallel program as to how fast you can go. And if you are going to do this, don't measure, don't actually compare your time on 1 processor, 2 processors, 4 processors, and 8 processors. Just go for as many as you have. Whereas if you actually, want to be efficient, then figure out where sort of the knee in the curve is and pick a reasonable number of processors. Also, as you'll certainly learn this afternoon, it's very easy to make mistakes while setting up a simulation. So you can easily set something up, let it run for several weeks, then look at the results, figure out what your error was, go back, and waste several weeks at a time this way, rather than simply going in, doing a short run, making sure everything's okay, then extending it to a long run. And if you get a NAMD output file, which 90% of the lines in that file begin with the word warning, if you don't understand those, don't worry about that, because we just print those out for our own entertainment. Finally, with all respect to the person in the audience who's doing the silicon simulation, NAMD is good at some things. It can be adapted to other things. If you wish to simulate ideal gases, or if you want to do very large solid-state physics simulations, those fields have their own specific tools, which have made different compromises and different judgment calls than we did when we were designing NAMD to do biomolecular simulations. So rather than leaping at NAMD because it's the first tool out there that might do what you want, look around and see what else is out there.
And finally, this is sort of in jest, but you can waste a large amount of time playing with your computers, setting them up different ways, experimenting and learning through trial and failure. And it's also a plug for one of Nandi's other strengths, which is running on commodity clusters, Linux clusters and the like. And next week, you'll actually have the opportunity to configure a Linux cluster based on our experiences, which is appropriate for running Nandi. And we'll also have a talk in which we'll discuss how we designed our clusters. We're now in our 3rd generation of Linux clusters in the group. They're quite affordable, and Nandi does reasonably well on them considering the price. So with that, I think I'll turn you over to Justin, where we can get back to a little of that good old lutefisk. Not lutefisk? Perhaps it's not.